Hi, everyone. It is my great pleasure to introduce Jeremy Baskin, the recipient of the ASBMB 2020 Walter A. Shaw Young Investigator Award in Lipid Research. Jeremy obtained his PhD in 2009 with Caroline Bertozzi at UC Berkeley, where he invented copper-free click chemistry and applied these selective tagging reagents to image membrane-associated glycans in vitro and in vivo. He then completed a postdoc in 2015 with Pietro De Camilli in the Department of Cell Biology at Yale School of Medicine, where he elucidated mechanisms regulating activity of a phosphatidylinosyl 4 kinase at the plasma membrane, including discovering implicating the kinase and its regulatory protein in an inherited disease of the brain's white matter. In 2015, Jeremy was selected to fill an unusual joint appointment at Cornell that bridges its Department of Chemistry and Chemical Biology to the interdisciplinary Weill Institute for Cell and Molecular Biology. After nearly 4 years on the Cornell faculty, Jeremy has established a major and vibrant presence in both his academic homes while cultivating numerous cross-campus research collaborations. Jeremy's independent research program includes several projects at the interface of chemistry and biology to study biological lipids and membranes. Work in his lab encompasses both methodology development and hypothesis-driven mechanistic research aimed at shedding light on how lipid properties such as structure and subcellular location impact biological function. Of his many important scientific contributions, I would highlight one that has been particularly important for lipid research. His group has developed small molecule probes to image production of the lipid second messenger phosphatidic acid by phospholipase D enzymes. Using this method that his group named IMPACT, they revealed several new regulators of PLD signaling, and I'm sure you will hear about those in his lecture. To date, Jeremy's contributions in this project area and his future promise has been recognized by several awards, including a Beckman Young Investigator Award, a Sloan Research Fellowship, an NSF Career Award, and a selection to Biochemistry's Future of Biochemistry special issue, an invitation to write a review article and accompanying technology feature on the chemical biology of lipids for trends in biochemical sciences. There is no question that Jeremy is a rising star in lipid research. His group uses a unique combination of chemistry, biology, and imaging. I am as excited about his future work as I am about him receiving this prestigious award. Without further ado and delay, Jeremy, please take over and present your lecture.
Hi, my name is Jeremy Baskin, and I just wanted to begin by thanking the organizers for the invitation, to thank the ASBMB for the tremendous honor of receiving this award, and to thank Thomas for the wonderful introduction. And also, before I start with the science, I would like to give one more thank you, which is to all of the amazing researchers, scientists, postdocs, students, past and present in the lab who have enabled all of our research. Here's a picture of us arranged as one of our favorite lipid-containing structures. So again, today I'll be telling you about our work on developing probes for lipid signaling. Why don't I begin by giving just sort of a broad introduction to lipids? If you don't think about lipids every single day, if you think about them in terms of a biological context at all, you might be aware of the fact that they're really good energy stores. They're good materials for storing energy. And so that's why we like to eat foods that are lipid rich, and why when we have excess nutrients in our body, we store that as energy in the form of lipids. And from the macroscopic to the nanoscopic level, lipids are incredibly important molecules as components of membranes, which are the barriers that separate the inside and the outside of our cells and of the organelles inside of our cells. And it's their bulk physical properties that enable the formation of membranes and and a lot of their critical functions. But beyond these sort of macroscopic properties, lipids are also really important because of their molecular identities. Lipids can act as signaling molecules, and they're hydrophobic molecules, and that's what defines them as such. But we can think of them just as important metabolites that happen to be hydrophobic, and in most cases embedded within bilayers within the cell. So on this slide, I've shown structures of several lipid molecules, privileged structures, most of them quite low abundance, that they all have hydrophobic portions on the bottom and polar head groups on the top, that cells use as signaling molecules to affect particular signaling pathways. And one of them, phosphatidic acid, will be the focus of today's talk. So just by way of general introduction, what I mean by signaling molecules is in the context of how cells are able to communicate in a multicellular organism by sensing cues from the environment, such as proteins, peptides, hormones, and small molecules that are sensed by receptors, whose activation leads to the accumulation or production of privileged second messengers, famous ones such as soluble second messengers, such as calcium and cyclic nucleotides are excellent examples. And certain privileged lipid structures, such as some of the ones I showed on the previous slide, can act as second messengers. And really one of the key mysteries about second messengers is how there are relatively few of them, but so many different potential downstream effects that can occur downstream of their production. And so the information that allows cells to go, through the pathway of sensing different combinations of extracellular signals, and going to the right physiological response, whether it's change in gene expression or cytoskeleton rearrangement or membrane trafficking or signaling response. That information is encoded within the molecular identity of the second messenger and also the spatio-temporal context in which it's generated. And so those challenges of understanding how lipids can cause specific signaling outcomes really motivate our work. And the types of questions that we're really interested to try to answer are ones that largely have motivated us to develop new tools. And these are wanting to know at the organelle level, where are the key signaling lipids in the cell produced downstream of specific stimuli? And beyond knowing the identities of the enzymes that are responsible for their production, how do cellular networks regulate the activities and the localizations of those enzymes? And then as a consequence as a sort of flip side to being able to visualize where the lipids are, what are the consequences of producing lipids on one organelle membrane or another? So in other words, a lot of things happen when cells choose to make a particular signaling lipid. And in our lab, we've really focused on two main classes of lipids. They're both phospholipids, they're both anionic, and they're both fairly low abundant. The phosphatidic acids and the phosphoinositides, they share in common the glycerol fatty acid backbone, and what's different is the anionic head group. And in today's talk, I'm going to focus on the chemical biology side of the work in our lab, which is focused on phosphatidic acids, these enigmatic, simple phospholipid structures. And in order to answer some of those questions that I've posed, we've really had to dive in and develop a lot of new chemical biology methods, and I'll summarize some of our progress in the next several minutes. Let me begin by telling you a little bit about phosphatidic acid. I think it's a really interesting lipid. It might be describable as enigmatic. It certainly is pleiotropic because it can cause many different signaling events to happen. First things first, phosphatidic acid is a biosynthetic intermediate, and certain enzymes in the endoplasmic reticulum synthesize it en route to other phospholipids and other lipid species. But its levels are kept low because it can also elicit signaling. But phosphatidic acid pools for signaling are generated by different enzymes downstream of cell surface receptor activation. And those pools of phosphatidic acid can change membrane properties, they can recruit effector proteins, and the net result is that you can have many different effects at the subcellular and the cellular and the tissue level as a result of phosphatidic acid signaling. And so the key question that sort of drives all of our work is, how does a single molecule, or more accurately, class of molecules with different acyl tail forms, but where the business end, the polar head group, is the same, how does this single type of molecule mediate so many different biological functions? And again, it's the spatiotemporal context, the location that is produced, and what else is in that environment. And so the types of questions that I'm going to address in today's talk are really the broad ones that I formulated a few slides ago, but focused on phosphatidic acid. Can we develop tools to see where it's made at the subcellular organelle level? Can we apply those tools to understand how cells regulate its production with greater precision? And what are the consequences of making phosphatidic acid on one membrane or another with respect to signaling events? So today I'm going to tell you about two complementary sets of tools to visualize and tools to perturb phosphatidic acid production in cells. And I'll give you some glimpses into some of the applications that we've undertaken to dissect these roles. Okay, so let me highlight some of the students in the group who have really pioneered this work. And the visualization tools were pioneered by two outstanding students in the group, Tim Bumpus and Dongjun Liang. And so let me tell you first, the enzyme class that we've focused on the most that synthesizes phosphatidic acid, and these are enzymes known as phospholipase D, or PLD enzymes. Okay, so PLD enzymes synthesize phosphatidic acid through hydrolysis of phosphatidylcholine, which is the most abundant phospholipid in all of our membranes. So we wanted to be able to visualize not all of the phosphatidic acid in the cell, but just the phosphatidic acid produced by PLDs, which is downstream of important signaling receptors. And in order to accomplish this task, we took advantage of a really important second reaction that PLDs are known to catalyze, which is called transphosphatidylation. So if you feed cells primary alcohols, such as ethanol, PLD enzymes will use them as a nucleophile instead of the natural nucleophile water to produce alcohol-containing lipids, where this alcohol is incorporated into the head group. These are called phosphatidyl alcohols. And this is a really impressive reaction. So first of all, if you radio label all of the lipids in the cell with, say, C14 labeled palmitic acid, then you'll get different spots of different migrations on a thin layer chromatography plate. And if we feed cells ethanol when the PLDs are active, we'll get a new spot, and that's the phosphatidyl ethanol. This is also commonly done with N-butanol. So what's really amazing about this reaction is that you can add tracer concentrations, micromolar, low millimolar concentrations of alcohol. And in the presence of 55 molar water, these PLD enzymes actually prefer the alcohol substrate and make substantial amounts of the phosphatidyl alcohols. The tragedy of this classic assay, which in more modern contexts has been adapted to stable isotope labeling and mass spectrometry analysis, the tragedy is that these phosphatidyl alcohol lipids are made in cells at particular organelles where the endogenous PLD enzymes are active, but they're detected in bulk biochemical measurements outside of the cell. And so we lose that spatial information. So we reasoned that if we could put a spectroscopic tag directly onto the lipid itself as a function of PLD activity, then we could image their locations in cells. And that's the idea for a tool set that we're calling IMPACT, for imaging PLD activity with clickable alcohols via transphosphatidylation. So we replace the natural the natural commonly used probe, ethanol or butanol, with a synthetic non-natural alcohol that has a small red group on here. I'll show you structures of those in a moment, a clickable group or a bioorthogonal handle. So this is small enough that the PLD enzymes will still accept this as a transphosphatidylation substrate, but it can be tagged with a fluorescent molecule like a BODIPI or a fluorescein using a very selective chemical reaction called a click chemistry reaction. And so if we can carry out these two steps rapidly, then the location and abundance of these fluorescent lipids reports on where these PLD enzymes are naturally making phosphatidic acid. So this strategy works remarkably well with a number of different alcohols. We've used alkyne-containing alcohols, including an alkyne-containing analog of the head group of phosphatidylcholine. And these can be detected with azidofluorophores by a copper-catalyzed azide alkyne cycloaddition, which is a form of click chemistry. This works really well in fixed cells, but the copper catalyst is toxic. So in order to visualize these lipids in live cells, we also use azido alcohols. And PLD enzymes will accept a variety of primary azidoalcohols, and these can be detected with strained alkynes known as cyclooctines in a strain-promoted click chemistry reaction. So what kind of information can we get from these assays? Well, let me illustrate it with an image, and I'll sort of describe a lot of the control experiments that we've done. First of all, IMPACT is an activity-based imaging approach. So what we're looking at is a single cell, and the fluorescence is not all of the phosphatidic acid in the cell, and it's not all of the PLD enzymes, it's just a subset that is active. And this is fundamentally different information you get from a protein-based imaging probe that will bind to all of the various lipid targets. We can do co-localization experiment to look at the organelle locations, and we do all sorts of validation with inhibitors and Crispr knockout and with mass spec and HPLC analysis to make sure that what the fluorescence is corresponds to the expected molecules. So this was really exciting for us to be able to visualize where the locations of the PLD enzymes were that were making phosphatidic acid. But we were a little puzzled by the endoplasmic reticulum localization, and we were also puzzled by the lack of a plasma membrane localization, which is a place where PLD enzymes were known to act. And that got us wondering whether our labeling method was fast enough because there are other competing processes that can traffic lipids around the cell. And so the analogy, you know, we're at Cornell University, we're a big hockey school, and I grew up in Canada. The analogy is illustrated well, I think, by this photograph. The photographer had to be just ready and with very high temporal resolution to be able to snap that shutter before Bobby Orr falls to the ice after celebrating his Stanley Cup winning goal. And so we wondered whether the IMPACT tagging labeling reactions were occurring fast enough because lipids can diffuse within membranes and traffic to other organelles. And if the tagging reactions take too long, then what we're imaging is not where the enzymes were first active. So in order to get around that problem, we developed another more complicated, slightly, chemical tagging a set of reagents for a real-time IMPACT that uses a bulkier and more challenging to synthesize substrate, but one that engages in a really rapid click chemistry reaction known as a tetrazine ligation. And you know, so it's a big ask for the PLD enzyme to replace its natural substrate water with this gigantic, monstrous alcohol. But after searching and synthesizing a whole bunch of them, we found that this oxo-TCO alcohol, first developed by Joe Fox's group at Delaware, was appropriate for this application. And so by having a fluorescence turn-on click chemistry reaction that is very, very rapid, we can watch the click chemistry step happen in real time and visualize fluorescence without having to wait until we rinse out the excess click chemistry fluorophore reagent. And then we can capture these lipids just as they get tagged and then watch their behavior in real time. So in this movie here, which is 1 minute of the click chemistry reaction, which will loop over and over again as I talk here, what we can see is that when we stimulate cells with a 4-bol ester to strongly stimulate the PLD enzymes and add this TCO alcohol, this is a very short reaction. And then we turn on the microscope acquisition for a time-lapse movie, and we add the fluorescence turn-on click chemistry reagent. And what we can see from this movie is, here, I'll pause it right after it begins. At the very early time points, like 9 seconds, there's only fluorescence at the plasma membrane, which is very satisfying because that's where 4-bol esters should stimulate PLD activity. But then over the course of the next minute, as the fluorescence increases, because the half time for the click chemistry reaction is about a minute, we can start to see more intracellular fluorescence, first in the endoplasmic reticulum and then at the Golgi apparatus. So this is really exciting for a number of reasons. We sort of get an extra piece of information. If we look at this early time points before the fluorescent lipids have a chance to move about the cell, this is telling us where the PLD enzymes are active. And then as we watch those lipids over time, they are revealing how they get moved around the cell by the machinery that moves phospholipids from one organelle to the other. So if we label the ER in a different color, we can track the internalization from plasma membrane to ER by the increase in the co-localization as measured by the Pearson coefficient. So this is a way to track plasma membrane to ER trafficking. And what's really consequential and important about this result is that that is a newly appreciated way by which the bulk of phospholipids are transported around the cell through transport at membrane contact sites between the endoplasmic reticulum and lots of other organelles. And this stands in contrast to the vesicle transport pathway, which has much slower kinetics. Now, the non-vesicle pathway, there are not a lot of good tools for visualizing this kind of transport. And so sort of like we buy one, get one free, we develop IMPACT as a tool to look at phospholipase D signaling, but it also is proving to be really useful at tracking bulk phospholipid transport between the plasma membrane and the ER. And at the end, I'll return to some applications we're working on to look at ER transport between ER and other organelles. So getting back to looking at the PLD activity, we then replace the formol ester, which is a very strong stimulus, with physiological stimuli of G protein coupled receptors and receptor tyrosine kinases. And then if you just look at the 9-second time point of those real-time IMPACT movies, we can see where the PLD enzymes are that are activated downstream of those different physiological stimuli through the activation by different intracellular intermediates. And from looking at these two images, which are representative, we can see plasma membrane localization downstream of this GPCR and intracellular localization downstream of this receptor tyrosine kinase. So at this point, these studies have been published, and I want to highlight in the next couple of slides some unpublished work that highlights some of the applications and directions that we're going with some of these tools. So in one study, so as an overview before I get to that, I just want to sort of highlight what IMPACT is capable of. Again, it's this two-step chemical labeling, and it can reveal not only bulk population, but also at the single cell and at the subcellular level using these different analytical techniques, the activities of phospholipase D mediated phosphatidic acid signaling. So as I was mentioning, let me tell you about a couple of applications, and then I'll move on to our other final tool. One application that we've looked into is how to use IMPACT as a readout of the GQ branch of GPCR signaling pathways. And we focused on this really interesting GPCR, the parathyroid hormone receptor, which signals both through GS and through GQ. And through GS, it has this really interesting non-classical signaling where it has persistent cyclic AMP generation from complexes of the PTHR with beta-arrestin and GS that are on endosomal surfaces downstream of internalization, which is in contrast to most GPCRs, which have their signaling turned off by beta-arrestin mediated endocytosis. So a lot of really good tools for looking at cyclic AMP have revealed the GS side of this pathway, and we have realized that downstream of the GQ phospholipase C pathway is the activation of PLD enzymes. And so we are using our real-time IMPACT reagents to look at the localization and the duration of the GQ branch of this parathyroid hormone receptor trafficking, which has important physiological consequences for developmental processes and organ functions that require the GQ branch of PTHR, and some of which can be disturbed in diseases. So that's an application of IMPACT to look really at the subcellular level at specific signaling pathways, and more generally as a tool to look at the GQ branch of GPCR signaling. Another application that takes advantage of single cell labeling is another unpublished study that we're currently working on, which is to use IMPACT as a readout for the for a genome-wide CRISPR screen. And so just as a sort of brief orientation, CRISPR technologies, including CRISPR interference, but other ones as well, have been really useful as ways to interrogate specific phenotypes and connect them to new genes and to discover functions of genes and relate them to pathways. So in a particular genome-wide screen, cells will be engineered using lentiviral transduction to have a guide RNA that targets a single gene to inactivate a single gene, say in CRISPR interference, by preventing its transcription. And then typically there's a phenotypic readout, a GFP reporter of transcription, or antibody labeling and internalization, or a live dead assay. But we realized that we could use this chemical biology approach to fluorescently label cells downstream of specific PLD signaling activities, and then sort the cells by fluorescence activated cell sorting to have the high and low cells, which are the outliers on the histogram, report on genes which would be, when knocked down, causing the cells to have higher or lower PLD activity. And so these would be, from the screen, putative inhibitors or activators of phospholipase D signaling. And so we've done some of these screens, and we're starting to look through and validate several of the hits that are in them. And I think they point to a really important application of chemical biology fluorescent labeling tools to discover functions for new genes in important, precise, targeted signaling pathways. So those are some of the applications of IMPACT, our tool for visualizing PLD activity. In the last few minutes, I want to briefly highlight some of our complementary tools to manipulate or perturb phosphatidic acid production. And the basic motivation here is, okay, so what if you can see that the lipid is in one place or another? I would like you to show me that making it in that place is causal for the downstream signaling events. And this is a challenge that Rekha Te, a really fantastic student in the group, has taken on. So the thought experiment is, what if you could flip a switch and make phosphatidic acid only at the plasma membrane, or only at the endosome or some other organelle? Spatially controlled phosphatidic acid synthesis. So the tool that Rekha developed in order to do this is a light-controlled or optogenetic phospholipase D. So we call it Optopld. And the way that it works is that we took an enzyme, this PLD, and we hooked it up to Cry2. Cry2 is a protein that can heterodimerize with CybN in the presence of blue light, but not in the dark. And CybN is tethered to a peptide sequence that will localize it to one or another membrane location, say a transmembrane sequence from a membrane-resident protein. And so in order to use a good PLD, we want one that's not subject to regulation, like a mammalian PLD. And so we found a bacterial PLD from Streptomyces that is soluble and has activity in the mammalian cytoplasm, but is sort of like a dumb enzyme, and it just sort of hangs out in the cytosol where it doesn't have any lipid substrates. And when we shine blue light, this fusion protein goes to membranes and makes phosphatidic acid. So first, let me show you that the tool works. When we put the fluorescent tag on the PLD itself, and we put CybN at the plasma membrane, the PLD is in the cytosol, and it moves to the plasma membrane. When we put CybN on other organelles, when we shine light, the PLD goes to those organelles. Using IMPACT, we can quantify by flow cytometry that when we shine light, there is a large and equivalent amount of PLD activity on various organelles, but in the dark, there's much, much less, and that activity depends on the catalytic residues being intact. So what type of applications can we do with this Optopld system? We've looked at this signaling pathway called the Hippo pathway, wherein it's a growth restriction pathway. These kinases in the cytosol phosphorylate this important transcription factor and prevent it from going to the nucleus, and that prevents proliferation. In the presence of various stimuli, including, as recently reported, phosphatidic acid, the Hippo signaling is turned off, the transcription factor YAP goes to the nucleus, and cells grow and divide. So it's really important to try to understand what are the localizations of the phosphatidic acid in the cell that allows this Hippo bypass and proliferation to occur, as a first pass towards trying to understand what are the exact PA effector proteins. So we wanted to validate whether this report was true, and then use our Optopld to establish which organelle pool is important. So the readout is immunofluorescence for this transcription factor YAP. In the absence of phosphatidic acid, it's in the cytoplasm and not in the nucleus. And when we add phosphatidic acid to cells, as was done in those published studies, the transcription factor moves to the nucleus. If instead we make phosphatidic acid not by adding it to the media where it can get internalized and go everywhere, but using Optopld, we precisely make it on one organelle or another, we find that making equivalent amounts of it at the plasma membrane relative to controls results in much more translocation of the transcription factor to the nucleus compared to making equivalent amounts in other organelles. And so this establishes the plasma membrane pool of phosphatidic acid as the key pool for suppressing Hippo signaling. And this is a first step along the way towards understanding the PA effector proteins that are responsible. The final piece of data I'll show you is that we've been able to combine directed evolution with impact in order to evolve mutant versions of the Optopld that have different catalytic processes, catalytic properties. And this is using a standard in-cell directed evolution platform using yeast that are transformed with libraries of PLDs where there are mutations made by error-prone PCR. And using this platform, we've identified a number of single mutants to PLD. Their PLD activities, as determined in vitro assays, are on the x-axis relative to wild type and catalytic dead. And what's really amazing is that when we put equivalent amounts of those into cells and look at the hippo pathway readout, there's an exquisite sensitivity to the dose of phosphatidic acid exhibited by cells when we look at the amount of this transcription factor YAP that goes to the nucleus. So it tells us something about how we can really titrate the effect of hippo signaling by how much PA that cells have within them. And so some of the things we're doing with our Optopld tools are combining them with other types of tools to elucidate mechanisms and discover PA effector proteins in hippo pathway, combining them with impact to make fluorescent lipids at particular locations to look at intracellular lipid transport, and combining with other types of alcohols to engineer unique properties into membranes for synthetic biology applications. So in summary, I've told you about these two tools: impact for visualizing phospholipase D signaling and Optopld for controlling phosphatidic acid signaling. And I have only left to thank all of the really amazing students in the group who have done this work. I've highlighted the contributions of the 3 graduate students. I've also in red here mentioned the names of undergraduates involved in this work and our collaborators on some of the projects. I'm very grateful to these agencies for supporting our work, and to the ASBMB for the honor of this award and for the opportunity to share everything with you. Thank you so much.